All right. Good day, everyone, and a warm welcome to this webinar. I'm Carrie Zhang. It's a pleasure to have you with us for this session on engineering microbes and microbial communities for sustainable bioproduction. Before we begin, let me briefly introduce our host, Bulk Sciences. As a premier end-to-end CDMO, Bulk Sciences specializes in advanced fermentation development and manufacturing, serving the pharmaceutical, food, and cosmetic industries. Biosciences provides comprehensive services, including custom fermentation development, GMP manufacturing, and full regulatory support. For more information, please visit our website at boxscience.com. Today, we are honored to have Dr. Rodrigo Ledesma-Amaro with us today. He'll share his pioneering work in developing tools to design and control synthetic microbial communities that outperform monocultures in the production of foods, chemicals, materials, and pharmaceuticals. As we embark on this exciting journey, I'd love for you all to get involved. During the presentation section, you will be able to post your questions related to the webinar content using the Q&A panel at the bottom of the screen. So without further delay, let's give a warm welcome to Dr. Rodrigo Ledesma-Amaro. Dr. Amaro, the floor is yours.
Thank you, Carrie and Book Science for the opportunity to be here. Can you hear me well?
Yeah, very good.
Perfect. Great. So I will share my screen now.
Mm-hmm.
I think yeah, okay. Cool. You should be able to share my slides.
Yes.
Let's bring the pointer up. Great, thanks everyone for joining. And I'm going to be talking about engineering microbes and microbial communities for sustainable production. So let me start by telling you how I'm structured the talk. I'm going to be talking for the next 40-45 minutes, and then there's time for questions. I'm going to start giving you a bit overview on engineering biology and how microbes can be a solution for our manufacturing issues, but I'm sure that you all know about that, so I will be brief. Then I will tell you a little bit about the center I work for, Direct, which is the best center for sustainable protein. So you have an idea of how microbes and microbial communities can also play a role in changing our health, both human and planetary. And finally, I'm going to go deeper into microbes and microbial communities engineering for different products that we've been developing in the lab in the past years. So I'm a professor in engineering biology at Imperial College London, where I'm based, and the best center of microbial food hub are also based here at Imperial College. So manufacturing nowadays is heavily based on unsustainable practices and fossil fuel and chemicals coming from petroleum. So we need a manufacturing system that is more sustainable, secure, and healthy. So we're trying to move away from this kind of non-sustainable fossil fuel-based economy to what we call the bioeconomy, trying to make everything we might need from food, fuels, materials, etc., based on biological sources that are more renewable and sustainable. So in that context, there's this discipline that started around 25 years ago now and has been developing fast in scientific community, but also transferring to industry more and more, which is synthetic biology. Now has been, at least in the UK, renamed to engineering biology. So I'm going to just briefly tell you, just in case this is not your background, that synthetic biology is a scientific discipline that is trying to make biology and engineering discipline. So that's bringing some concepts specifically for electrical engineering initially to make biology more controllable, predictable, standardized, and modular. In that context, metabolic engineering is another discipline that started a little earlier than synthetic biology and was bringing to biology concepts for another engineering discipline, this is chemical engineering. So you can see an analogy of a chemical production plant with different elements and units and connected through different pipes and fluids go on and chain chemical structure from one to the next. And that's very similar to what happened in the metabolism of cells, where different enzymes are going to convert some chemicals into another, and there will be a metabolic flux that go through them. So through this analogy, metabolic engineers have been using this concept to improve yields and production. So in the same way that traditional chemical engineering has been combining electrical circuits and chemical plants to build infrastructure that can deliver chemicals at low cost. With engineering biology, we can think as well about how we bring these control elements from genetic circuits to ways to control metabolism and engineer it to really help us move to the bioeconomy. But now thinking not only about the process scale, but also at the individual cell scale, the microorganism that can do the job. So microbes have been considered microbial cell factories in this type of application, as their metabolisms can really produce a large variety of chemicals, and those chemicals can have application in the biofuel, chemical materials, food industry. Naturally, they do produce chemicals, but their goal is not to do so. They didn't evolve to do that. So they produce usually small amounts. And those small amounts that natural microbes produce are too expensive to be manufactured at the scale and cost that is required to be successful in the market. So we need to perform this engineering biology process to make those cells more efficient and producing those specific molecules that we then want to purify and use. And traditionally, synthetic biology has used the design, build, test, learn cycle to go about improving these microbial cells to the point that they can become really robust and strong at producing those molecules. So just going to briefly tell you what this DBTL cycle is and how we operate in the lab on a daily basis. When we want to produce a specific molecule, we start designing on the computer what kind of the genetic changes we need to do, which is the best organism we need to use. We're basically planning and design the next steps that happen in the lab. So build is the phase that happens in the lab where we basically combine DNA parts using assembly methods such as Golden Gate, and then we edit the DNA using technologies like CRISPR, and then that helps us manipulate cells, engineer the metabolism to then go and test in the next phase how much they are producing of the molecule of interest. And for that we use process optimization to maximize production, but also analytical tools to quantify what are the molecules that are produced and what are the byproducts that are made as well. And the last phase of the cycle is the learning phase, where we usually use systems biology data, transcriptomics or multiomics, and we can incorporate AI in this phase as well to really learn from our design to our test what has gone well, what has gone wrong, what is unexpected, how can we learn from biology by doing so, and more importantly, what new elements can we incorporate in the next cycle of the design phase to make the strains even better. And that cycle can go over and over until the strain performs in the way that is required for industrial bioproduction. One very important step in designing a bioprocess based on microbial cell factories is the microorganism. So choosing the right microorganism can really save years of research and optimization of processes. So that's one of the most important steps. And we can choose by a variety of different organisms. Some of them are considered conventional organisms, though that we have been engineering and using at scale for a long time, like E. coli or Saccharomyces cerevisiae, bakery yeast. And some other are non-conventionals, maybe isolated from nature, maybe really good at making some of the precursors or grow specific conditions or from specific feedstock, but maybe not so easy to manipulate, or we don't have enough knowledge about them to really understand how to bring them in the right direction. And that's where synthetic biology or engineering biology has been playing a big role and has been accelerating this transformation of non-conventional to conventional organisms by developing more efficient methods such as CRISPR or more advanced computational tools like genome-scale metabolic models, and also the decrease of cost of sequencing and so on has helped in this domestication of non-conventional to conventional organisms. When we think about the microbe, it's not only how the microbe behave in an isolated way or in a lab scale. We need to think this is going to go in a large bioreactor. It's going to be part of a process, and the process is not just the fermentation itself, but it's whatever happened before and all the different pretreatment and feedstock that we want to utilize to decrease the cost and to keep the process sustainable, but also the downstream process. And depending on the product and the molecule that we want to make and the purity that is required for the application, we will need to go through different downstream process steps that can be significantly expensive. So when we choose the organisms and the engineering strategy, we can think about how we can improve the cell behavior for all of the things I mentioned above. So we can think of this multi-level strain engineering, where the strain is not only good at making the product, but is good at maybe making the product from specific feedstocks that are treated in a specific way. And also that the strain itself can help with downstream process, maybe by engineering the secretion of the product to the media or reducing byproduct that can interfere with the downstream steps. So we can do strain engineering thinking about the process. So microbial cell factories, microbial bioproduction, microbial biotechnology, there are a lot of ways to talk about this technology. Recently, it has been renamed as precision fermentation. Fermentation has been around for millennia. Even before agriculture, we were already fermenting fruits and other animal-derived meat or milk as humans, and we're very familiar with this. It's already 1/3 of our diets, and consider a great way to bring functionalities to food in particular. But now with the new technologies and engineering biology developing, we can start doing what we call precision fermentation, which is engineering that particular microbial strain to make one specific molecule of interest. When this concept has been around really in the market even since the 90s, with vitamin B2 commercialized by BASF, or rennet for cheese making processes. The term precision fermentation is more recent, and we mainly hear about it in the past 5 years. And I'm just bringing up this in the screen because we just published the precision fermentation Wikipedia 1 month ago. So we thought that now with this publication, we are consolidating precision fermentation as a new scientific discipline, or at least renamed scientific discipline, but now open for everyone to understand and read about it. So please feel free to send feedback on the content of this page, and we will be happy to update it. I'm going to go very briefly on this. Yeah, but I wanted to give you an example of application. We're going to go then in the science, and you will see many of the molecules we're making. But this is kind of a helicopter view of one of the problems that microbial biotechnology and fermentation can solve, which is a food problem. And in that context, we launched last year the Bezos Center for Sustainable Protein that goes together with the Microbial Food Hub. So these two centers work together with the idea of making food more sustainable, healthy, tasty, affordable, and secure. And it's bringing this engineering, biology, and AI enabling technologies to the food science as an application field to address some of the challenges that we have in our food system. For example, with prices that are too high, or quality that are too low, or the acceptance of new foods, for example, those made with microbes. So we're working a lot with fermentation, precision fermentation, biomass fermentation, traditional fermentation, and how microbes can create new food. And the center itself is not only research, it's also about education. We just launched the first PhD program in alternative proteins in the world, with 50 PhD students started last month. Translation is very important. And this is general for synthetic biology, which is very applied. It's all about how we translate the science into the market through partnering with companies, licensing technologies, or creating startups. So we just announced last week a new commercialization pipeline and accelerator program in our center. And we also have these pillars of knowledge and network, where we know we cannot do this alone. We need to create network partners with different stakeholders that can help fulfilling the vision, which is very, very broad. So we have more than 120 researchers in the center, more than 40 different research groups, significant amount of funding, and a set of partners that are different institutions that work together with us. And this is some examples of people and companies we work with, and we're always looking for more collaborations. We are structured in research and facilities, and I think this is also very typical from engineering biology, trying to not only do individual research, but aggregate technologies, offering services to companies and researchers to really accelerate development of products. In the case in our center, we also very interested in having this real life impact, and we do that through what we call the impact units, where we work in commercialization, company creation, sustainability, education, policy and regulation, astronomy, public and consumer, and also the global south. So we're very happy to work with others and solving this important and essential challenge that we're facing with our food system. So if you're interested in working with us in any of the things that are in the screen, please reach out. You can scan this QR code, follow us in LinkedIn, or join our newsletter and community. Great, and now I'm going to go deeper into engineering biology, microbial communities. And I will start by introducing my research group, who are the people that are really doing the work that you're going to see. So they are behind the scenes pipetting and creating this great science that you're going to see. This is a bit old picture now. Need to update that. And we are around 40 people at the moment in the lab. So you have already seen in my slides and images of the process. The typical bioprocess, where there's a feedstock that is fed into a microorganism in a bioreactor. The microorganism is going to convert that through the metabolism into a product, and the product is going to be purified, isolated, formulated, etc. The other element of this process is the engineering biology tools that allow us to domesticate microorganisms and to engineer them in a faster and better way. So I'm going to talk about these four elements as I go with my presentation. And this is, again, a helicopter view of the many projects we are working on in the lab, and I will be happy to expand on any of them that caught your attention during the questions. Feedstocks are super important in bioprocess. They contribute significantly to two essential things. One is the economy of the process. The feedstock need to be cheap, need to be affordable, need to be available, but it also contributes strongly to the sustainability of the process. And at the end of the day, the reason why we are doing bioprocesses in most of the cases is because we want to move away of our less sustainable practices. So we need to make sure that the process is sustainable, and choosing a sustainable, renewable feedstock is going to help with that. So we have been working with a variety of different feedstocks. For example, sugars. Sugar are the more obvious one, especially exoses and glucose, and sucrose are the more widely used feedstock in fermentation. They have some drawbacks. They can become expensive. They can compete with food sources, which may have implication in specific regions. But organisms like them. They work really well. Pentoses, so C5 sugars, are less easy to utilize by many of the microorganisms that we use at scales. For example, baker yeast cannot consume xylose naturally, or other organisms I will be talking about cannot consume xylose. What we can do, and we've been doing, is engineering some of these organisms to really be able to expand the sugars that they can metabolize, that can consume as feedstock. So now we created organisms recently that were not able to use things like xylose, arabinose, or starch as carbon source, but after engineering them, they can do it, and they can do now cheaper or more stable bioprocesses using those. While sugar has a limitation, I think on the other side, one of the feedstocks that everybody agree that would be ideal to use in biotechnology is lignocellulosic materials and hydrolysates. They are very abundant, they don't have a lot of uses, and they usually consider waste from agriculture, for example. They are difficult to extract or to use by microbes, and they do it very slowly. So we can again engineer organisms to use these feedstocks, or more specifically the hydrolysates from these feedstocks, more efficiently to make products. And we've been doing a bit of work on that in different type of hydrolysates from different plants. Another feedstock that is widely available is food waste. We waste 1/3 of the food that we produce, and it will be great to be able to recycle that back into bioproducts. So things like waste cooking oil or vegetable waste are things we've been investigating and utilizing as feedstock for bioprocessing. Another example of if is industrial byproducts. So many companies produce byproducts that are usually valorized in a way or another, usually sold at very low cost for various applications. And examples of this is raw glycerol produced during the biodiesel manufacturing from vegetable oils, or some waste from food industry, from food manufacturing, actually, that can be produced, and that include a lot of leftovers from agricultural products as well, containing starch or fructose or sugars. So we've been working also on industrial byproducts as feedstock. Another interesting one is urban waste. Maybe that will not provide feedstock for a large population. If we're thinking about feeding or creating materials at large scale, but it's also available in big cities. And it would be great to valorize that currently doesn't have a lot of applications. The second kind of holy grail, I think, of feedstocks together with lignocellulosic materials is what we call the C1. And C1 is molecules that contain one carbon. An example of this is CO2. So if we will be able to fix CO2 and convert it into bioproducts by microorganisms, that would be great. Natural organisms using CO2 are usually slow, difficult to engineer. Sometimes it depends on light accessibility and so on. But can we engineer cells to use CO2 or CO2 derived molecules? For example, methanol or formate can be converted electrochemically from CO2, and then we can also use microorganisms to consume those methanol and formate, which are all the C1 products. So in my lab, we've been engineered CO2, formate, and methanol utilization by some yeast that naturally don't do it. And this is one example, and this is a yeast that I will introduce in a second. It's called Jarrobia lipolytica. Jarrobia lipolytica cannot consume methanol, as I say, methanol, which is this molecule here, CH3OH, that can be produced from CO2 in an electrochemical step, and but Jarrobia cannot use methanol. So we had to introduce a pathway to convert methanol into formaldehyde, and then Jarrobia is able to convert the formaldehyde all the way to CO2. And that process generates an NADH that can be transformed into energy in the cell. Now that doesn't contribute to producing more cells, more biomass or product. It's just contributing to energy generation, which is good, but it could be better if we can also produce our product of interest from methanol. So in order to achieve that, we had to integrate two pathways that are found in some specific yeast that can use methanol. So one is the RUMP pathway and the other one is the XUMP pathway. They have some similarities, but also different enzymes, machineries that need to be transplanted into Jarrobia to recreate those pathways. And what you can see here in the slide, and I don't want to go too much into detail, is that after integrating the whole pathways, you see high methanol consumption here on the right and growth here on the left. So one example of that, if you look at growth of the microorganisms with or without methanol, you can see how the engineered strain is able to grow much better, doubling the biomass that it produces just by adding methanol into the media. And this is true for two different engineering strategies that we tested. One is expressing the pathways in the cytosol of the cell. The other one is expressing the pathways in the peroxisome of the cell. We, of course, tested that the methanol is actually going into biomass by C13 labeling, and we calculated the metabolic fluxes of the pathways. But more importantly, we wanted to explore if methanol can now be used by this yeast Jarrobia to make a product of interest. The product that we chose was beta-carotene. Is an antioxidant and vitamin A precursor and a colorant widely used in the food industry. And we saw that by adding methanol to the engineered strain, we were able to increase 20 to 30% more beta-carotene. Great, let's move to the next phase, and this is about choosing the right microorganism. I mentioned this briefly before. choosing the right microbe is critical, is not straightforward, and is a big decision because it's going to really impact the speed at which you can engineer your process and your product. Just an example, thinking about metabolic engineering, I've experienced myself how specific yeast, maybe baker yeast, widely used and understood yeast with a lot of tools, can be engineered very quickly to produce specific molecule A. And by putting the pathway, the same genes in Saccharomyces cerevisiae baker yeast, we can produce, let's say, 10 milligram per liter. In the same cassette, we put it into, for example, another organism like Yerobia lipolytica. Exactly the same genes can produce 10 or 100 times more just from the beginning. So with no further optimization. So it really can save a lot of time because, as you may know if you work in this space, increasing an order of magnitude is a significant amount of work, and sometimes it's even very difficult to achieve. But let me go back and tell you more about the microorganisms we are working with. We work with a variety of different organisms, and I mentioned we work mainly with yeast. Our favorite organism is Yerobia lipolytica. I just introduced it in my previous slide about methanol utilization, but I will tell you a bit more about it in a second. We also work a lot with baker yeast, Saccharomyces cerevisiae. We also work with some bacteria like Pseudomonas, C. acnes, Vibrio nitrigens. Vibrio nitrigens is the fastest growing organism known. It can duplicate in 8 minutes, which is more than 2 times faster than E. coli. We also work with E. coli, but we also work with filamentous fungi, and examples of those are Fusarium and Rhizopus here. I said Yerobia is my favorite organism. There are many reasons for that, and I won't go into detail now, but it's an organism that is generally recognized and safe. It's actually naturally found in food, so it's safe to use for application even for human products or products that need to be consumed by humans. It's been developed quite fast in the past few years. We have a lot of tools to engineer it. We understand a little bit its metabolism. It naturally accumulates a lot of lipids. So and you can see here, these are the cells of Yerobia, and in green you can see lipid bodies. These contain lipids, and that's why it's called lipolytica, because it's also very good at consuming lipids. So in this electron microscopic picture, you can see lipid droplets that are outside the cell, and the cell is basically feeding out of these lipids and then bringing them inside, and that's what you can see here in this other picture. So in this cover from Nature Biotechnology, you can see Yerobia cells growing in a petri dish, and that's actually like showing how Yerobia is actually very good at biotechnology, right? Some of the reasons for that is Yerobia can grow at high cell densities. It can use a variety of different feedstocks, and as I say, it can be engineered, and we know how to grow it at scale. Second. Yeah, so based on that, it's not only me working in Yerobia, it's a big community and growing community in both academia and industry. Here you can see some examples of the application of Yerobia engineering. So it can produce protein, alcohols, lipid, amino acid, organic acid, terpenoids, and many others as well. But in industry, there are processes and companies working in many of these products too. So in the next part, I'm going to tell you a little bit about the tools we developed to engineer these microbes to maximize the conversion of the feedstocks into the products. And we can start here from the top left. We've been developing tools that are in the genetic engineering space, especially after the discovery of CRISPR. As in many other disciplines, CRISPR has really changed the way we manipulate biology, and I will give you an example of some multiplexing CRISPR methods that we have been working on. Other tools that we developed include electrogenetics. This is using electricity to trigger gene expression in cells, so we can do inducible bioproduction, for example. We also work with modeling, and of course, like everyone else, we use AI too. We work with genomic scale metabolic models. These are mathematical representations of metabolism that allow us to estimate or predict metabolic fluxes, helping us also design a new modification that we should be making in those microbes to change the production pathways in the way we want. And AI can become very useful once you generate data to kind of provide also some predictive information. We also work a lot in protein expression tools, and this goes through the development of new promoters that some of them can be inducible, new signal peptides that allow the secretion of proteins outside the cell, and also more increased secretion by engineering cell wall, cell trafficking, and so on. Something that we've been very interested in as well is in high throughput and automation, and how things like the biofoundries and robotics can accelerate the creation of new strains, and also how we can use high throughput not only to generate strain but also to screen strains and help with the analytic side of things, including bringing new single cell technologies. Link to single cell, something that I won't have the time to talk about today, but it's a very active research area in my group, is heterogeneity. And heterogeneity means in a cell population, you can have, when you look at what every single cell is doing, you can have a more dispersed behavior where you have cells that are really not producing and cells that are producing, or you can have a more homogeneous like cell population where all of them are behaving more similarly. Ideally, in a bioprocess, what we want is a homogeneous a population where all the cells are producing as much as possible of the product of interest. But that's not the reality. The reality is we have heterogeneity that is maybe reducing our potential yields. And so we're doing a lot of work in understanding heterogeneity in bioreactors, especially at large scale, and then designing cells that can fight that heterogeneity and reduce it. The other set of tools that we've been working quite strongly, and is actually part of the title of the talk today, is microbial communities. And I'm not going to spoil the presentation now, because later I have a whole section about microbial community tools that I'm going to tell to you about. But talking about tools, I chose an example to explain you the kind of things we are doing. And this is a CRISPR example. So CRISPR, as you know, is this technology, molecular technology that allows us to engineer genomes in a more efficient way. Usually, it works as a combination of a Cas enzyme, for example, Cas9 or Cas12, that binds an RNA, and the RNA helps this enzyme to find a specific region in the genome where the complex RNA and enzyme is going to have an effect. And that effect can be the cutting the DNA. It can be inhibiting gene expression, maybe by blocking the binding of RNA polymerases, or it can be enhancing gene expression by recruiting more of the machinery that increase bioproduction. So the part that I really like about CRISPR, and I think has a big impact in strain engineering, is the capacity to multiplex. And multiplex means that we can do multiple modifications in one single go just by having multiple guide RNAs being expressed. So typical metabolic engineering, we want to express several genes, delete several genes. In this example, there are 10 modifications. If you want to do one after the other, recover the market, start again, this can take 6 to 12 months. With our multiplex CRISPR, where we can modify more than 10 modifications in one single transformation, that can take 2 to 4 weeks. We can really accelerate the engineering space. So how our multiplexing CRISPR work is by developing a highly multiplex system to express many guide RNAs at the same time, up to 24. Then having two different Cas enzymes, one that is targeting activation, one that is targeting repression, so we can do both simultaneously. And also making the system inducible, so you can decide when the bioproduction phase really starts. And this is an example of application where we want to produce succinic acid. So this is a chemical building block. And we knew from the literature different targets that should be downregulated in red or upregulated in green. We created a guide RNA array with all these different guide RNAs targeting this different part of the metabolism. And then we looked at gene expression, and in one single transformation, we manipulated the expression of 11 different genes. And the gene that was engineered was able to produce several orders of magnitudes more succinic acid production than the non-engineered strain. Cool. Now I'll move to products. And so over the years, we've worked in more than 60 different molecules and enzymes that we produce at the relatively high levels in the milligram or gram per liter scale. And those include biofuels, chemicals, pharmaceuticals, materials, and also as well a bunch of different food and food ingredients that we'll be working on. Just examples of some of the things we've done, in particularly in Jarobia, include protein, lipids such as omega-3 fatty acid, cocoa butter, or human milk fats, or palm oil. We work on antioxidants as well, sweeteners, colorants, aromas, fragrances, flavors, preservatives, and vitamins. And that takes me to the next and last part of the talk, which is what about using microbial communities instead of a single microorganism? The typical process is one microorganism is converted the feedstock into the product. Now, when we look at nature, we don't find ecosystem where all the resources are used by a single organism. It's always a coordinated environment with multiple organisms specialized in different tasks, making the better use of resources. And that's how we envision the bioprocesses in the future are going to look like. And we're starting to see some early examples of this taking off, but you can imagine that instead of having one organism, we have multiple organisms where we have specialization and optimization of resources and recycling of byproducts, really maximizing yields and really utilizing fully those complex substrates I was mentioning before. But in order to get to that dream scenario, we need to develop tools to engineer communities. The same way we have been developing tools to engineer single cells in the past few decades. And some of the tools we need to create are intended to create division of labor, where there are cells specialized in different things, for example, splitting a metabolic pathway between two cells. Cross-feeding, where these different cells depend on each other because they produce a molecule that the other one needs, and vice versa. And that creates robustness, and that's something that is found in nature very often. And finally, control population ratio. So if we want to optimize a process with more than one cell, we really want to know and control how much of each populations are in the process to optimize the metabolic fluxes. So I'm going to give you very quick overview of some of the projects that we have worked on in the past couple of years. First, we had to develop cross-feeding mechanisms in Saccharomyces cerevisiae. And for that, we use Saccharomyces knockout collection. This is more than 5,000 different genes, so knockouts in individual cells. So you have 5,000 different strains of yeast, each one knockout in one particular gene. We selected 92 that are able to grow in rich media, but not in minimum media. What it means is they need something from the environment to grow. And then we put them together using robots in a pairwise combination to identify which ones, when they come together, they can grow, but they don't grow when they are not alone. And we found a bunch of them that could complement each other and create a community. You can see here all the high bars are the co-cultures and the individual bars are the monocultures. So only when they are together, they can clearly grow. And then we developed division of labor. This is a specific pathway producing a bioplastic precursor, malonic semialdehyde. It has an intermediate beta-alanine, but it's basically two enzymes making the product. So you can put, instead of putting two enzymes in one single cell, we can put one enzyme in one cell, the other enzyme in the other cell, create some cross-feeding behaviors, and see how much of the final product they make. And we found that the co-culture in some cases produce twice as much as the monoculture, the strain with the two enzymes in one. So that means in some cases, communities can outperform monocultures. So encouraged by that, we created a toolbox for population control in microbial communities, also in Saccharomyces cerevisiae. And that toolbox contained 15 different auxotrophies, 3 fluorescence protein, 15 exchange metabolites that could be combined to create not only 2 members communities, but three member communities as well. And we develop different strategies to control population in a more defined way as well. We tested this toolbox in another product. In this case, resveratrol. It's an antioxidant naturally found in fruit, but widely used for other applications as well. And in this case, there is 3 steps, 3 enzymes that need to be added to Saccharomyces cerevisiae to produce these molecules. And similarly, we produce a division of labor scenario, which one cell is pressing 2 enzymes, the other one is producing 1, or vice versa. And we tested this, and we found that in some cases, the division of labor and the cross-feeding produced 3 times more than the strain with the whole pathway in one single cell. So in some cases, division of labor really works. In others, it doesn't. So now that we proved that this worked in a conventional organisms such as Saccharomyces cerevisiae, we wanted to see if this also worked in our favorite yeast, Jarrobiella politica. So we did some knockouts, some auxotrophies, and we create some pairs. We put them together, and we saw that how when they are together, they can grow. When they are not together, they cannot really grow. And we were able to control population ratio as well by changing inoculation ratio and other mechanisms. And that led us to explore if we can create communities with more than one species. So can we create a community with a Saccharomyces cell and a Jarrobia cell? So that's what we did next, and indeed it worked. And you can see in some cases the monocultures don't grow, the co-cultures grow, not in all of the axotrophs, of course, and we were able also to control population ratio, so we can have scenarios where we have 95 to 5 of the 2 population, and others where they are more closer to 50-50. So we tested also a pathway to make a bioplastic precursor, in this case 3-HP, 3-hydroxypropionic acid, by doing division of labor and cross-feeding. And in this case, and this is difficult to see, but you can see the monoculture barely produces 3-HP, while some of the co-culture are really producing several orders of magnitude more. Same feedstocks, same process, just 2 strains instead of 1 in the flask. But we can use communities to do more than that. We can use communities that are bringing some pathways that are very difficult to engineer, such as CO2 fixation. So what we did in this project is put together a community of an organism, cyanobacteria, that can use CO2 naturally, and a Jarrobia that can make, in this case, beta-caryophyllene, which is a food flavor. So we engineer cyanobacteria to convert CO2 into sucrose, then Jarrobia to convert sucrose into beta-caryophyllene, and then combining the 2 together, we were able to produce CO2 to make CO2 and light conversion to beta-caryophyllene, which would be impossible to engineer in Jarrobiella politica alone. And the last example is how we can upcycle byproducts. So for those of you familiar with fermentation and fermentation-based products, the typical process always generate byproducts. So, for example, a cell that is consuming the substrate and making the product, it's also making something else we don't want. This is typically ethanol in Saccharomyces cerevisiae. It can be citric acid in Jarrobia, acetate in E. coli, and it's usually a way to balance redox in the cell or other metabolic perturbations. So what we created is a system based on a community of 2 cells that where one cell cannot use the substrate, but has been engineered to use the byproduct as a substrate. So these cells only going to grow. It's only going to grow if the cell of interest is producing byproducts, and it's going to upcycle this byproduct into more product. So we did that with Jarobia lipolytica. We engineered a strain that was able to grow on citric acid, which is the main byproduct, and not consume glucose. When we put them together with a wild type strain, the biomass increased twice, so double the biomass just by having a community instead of a single cell. And we tested this with bioproduction as well. Again, with beta carotene, we were able to produce 2 times more beta carotene than the monoculture, both in minimum media and lignocellulosic hydrolysates. This is just very brief because it's been just accepted, not yet available, but you can get early preview of this new work from the lab, and is how we can actually use cell-cell addition and spatial configuration to improve bioproduction. And going very quickly, we develop a toolbox to bring cells together in space that help them exchange metabolites in a more efficient way. So if they are doing division of labor, they can transfer physically those materials more easily. We can create patterns, we can create different configuration in space, and that at the end of the day help us also build communities that can produce 30-40% more production levels than the community without the seal addition, which is already producing more than the monoculture. Great, so with that, I'm finishing now. I remind us all why we are doing fermentation, because we want a manufacturing system that is more efficient, more cleaner for the environment and for us. But if we are not careful, we might end up over abusing the resources of our planet and needed to go outside our planet to survive. And if we do that, the good news is that fermentation can also play a role and produce the food, the materials, and fuels that we need to survive in space, in different planet, in a colony, or in a spacecraft, and without needing to bring all these elements from our planet. So a few months ago, we tested this and we launched some of our engineered yeast strains producing biofuels, chemical foods, and materials. And they were flying around our planet in order to start investigating how we can make fermentation happen outside our planet too. We actually were launching with SpaceX and feature in BBC, where we actually use microbial ingredients from the cells that were sent to space to create some food that the first British astronaut tested in our kitchen, and she said that it's much better than the food the astronauts currently eat. With that, I would like to thank all the lab members for the hard work, the funding agencies for supporting our research, Box Science for the invitation to be here today, and all of you for your attention. And I will be happy to take any questions.
All right, thank you very much, Dr. Amaro, for such an insightful presentation. And we will now move on to our live Q&A session. Let me check. Yeah, maybe Dr. Amaro, you can address the question from the panel, like one by one.
Let me read the question.
Okay.
I am Krishna, Laboratory Aliveda from Italy. Since you have extensive experience with Jarobia lipolytica, could you please explain the key parameters required for its in vitro cultivation? Additionally, does Jarobia lipolytica secrete or incorporate any vitamins or short-chain fatty acids similar to Saccharomyces cerevisiae? I guess the first question is about the key parameters. So Jarobia is an aerobic organism, so it requires oxygen to grow. I think that's a key element when you think about how you work with it at scale. So it needs a good supply of oxygen to grow efficiently. It can grow in a variety of feedstocks, as I mentioned before, and it can be engineered to use even more. It's quite tolerant to pH. It has a wide pH range, can grow a low pH quite nicely. It has a relatively wide range of temperatures it can grow as well. It can grow quite okay at lower temperatures, and some strain can go up to maybe 37, 35 degrees without evolution. You can push that with evolution. And yeah, it does. It can use both minimal medias and advanced media. It can use alternative nitrogen sources like urea, for example. The second question is about secreting or incorporating vitamins and sorting fatty acids. It can, yeah, if you add vitamins, it's going to incorporate them. Fatty acids as well. Any type of fatty acid, Jarobia is going to like to incorporate them. We engineer Jarobia to secrete fatty acids, so that's something that can be done as well to favor the downstream process. There's another question from the same person. How stable are its bioactive compounds after spray or freeze drying? Actually, that's a great question, and I think if I can show you one example, I'm just in my office and I have some lyophilized Jarobia here. So this is from a company in Poland that they commercializing Jarobia products. So I believe actually checking the expiration day, uh, this has been bought over a year ago, and you have Jarobia that is dried and stable, and in this case, providing probiotic activity. So I believe it can stay for quite a long time with bioactive compound. But I guess it also depends in the compounds themselves. The other question is, could Jarobia politica be used as a based biomass for selenium or iron fortification? So yeah, I'm glad you asked that because that's something that is actually in these examples that I just showed you. And yes, and we work a lot with iron in particular. It's one of the biggest deficiencies in the UK, but in many other places in the world. Calcium and iron are still big deficiencies, as well as vitamins. So we are very interested in increasing vitamin content and secreting vitamin for precision fermentation, but also as a food supplement.
Thank you, Dr. Amaro. I noticed that Joseph asked if we can get the link to join your community. So maybe we can send the link later after via email. Okay.
Yeah, I can do that. And I think there's another a comment that is about what are the main challenges of C1 utilization. So I think there are several challenges, and depending on kind of the two approaches. One is can we use organisms that already use C1? Like I put the example of cyanobacteria, but there are other systems like Clostridium and others out there. They have challenges. Sometimes they only work in strict anaerobic conditions, which can have challenges from the kind of bioprocess design and bioreactor design. In some cases, they are difficult to engineer compared to other organisms. So preventing to do really heavy metabolic engineering type of experiments with them. In other cases, I mean, the one that need light, so they're more complicated to scale potentially, or if you do open tank reactor, they can get contaminations. I guess the other challenges on the other side is when you want to make an organism that is already quite good in bioprocessing, and you want to expand its feedstock utilization capacity to use C1s, like as I said, CO2 forming methanol. This is very exciting area of research in my opinion, and we've seen really scientific breakthroughs coming from different labs in the past 5-7 years. So it's an area that we expect to see much more development coming through. We are already seeing some companies emerging with these concepts in mind. It's still, I think, early days because we have strain that can use methanol, can use formate, can use CO2, but really at very low efficiencies, just to be able to go to large scale. Communities can play a role, I think, because we have organisms that can use CO2 and organisms that we can engineer massively, and then we can combine them. But it's also a lot of challenges because, as I say, community engineering has just started as a kind of field. Okay, there's another question from Carlos. Well, how is current legislation impacting use and development of products produced with engineered microbes? That's a good question. It's really country dependent. Different countries have different regulations. The US being more open, EU being more strict, the UK trying to transition to being in Europe to be more less strict, while keeping safety. In general, even in Europe, genetically engineered organisms are okay as long as you purify your product and in your final product you don't have any DNA from the organism that has been modified or any living organism. So as long as the separation is good, your product is not GMO. It's not GMO labeled, even if it has been made by a GMO organism. If you want to kind of eat or use the whole organisms with the engineered yeast and enzymes inside, then that's going to be a GMO product. And that's, I believe, very difficult to go through. And I don't think it has happened really in Europe yet. Although Impossible Burger, which is approved in the US, is doing this, and they're trying to get approvals in Europe and the UK at the moment as well. So that might happen, but it's much more complicated process than a specific product.
All right. I guess that's the last question, right?
I guess so. I don't know if everyone have access to the chat. I can put just there yeah, I think I cannot post in the chat. I'm trying to.
A link to the community, right?
Yeah, I just put in the link to
Yeah, you can put that in the chat. Yeah.
Yeah, for everyone. Sorry, he was going to the host only. Yeah, this is the website of our center, so you can go there and register from the mailing list in the bottom part, and then connect with us. And my email is available everywhere online, so you can find it and email me as well.
All right. So thank you very much, Dr. Amaro, for sharing your expertise, and to all of our attendees for your thoughtful questions. And today's discussion shows how engineered microbial communities can drive more efficient sustainable manufacturing. And as the team at Biosciences, you can count us as your trusted CDMO partner, and our fermentation development expertise can help move products from concept to scale. And our scalable infrastructure ensures a smooth transition from R&D to production. And our goal is to turn your innovative research into real-world solutions with end-to-end support. So for more information, please reach out to the team. And thank you again for joining us. And please follow us to stay updated on upcoming webinars. And wish you a nice day ahead. And if you have any further questions and anything, you can also reach out to us, and we will contact Dr. Amaro to address that. Okay. Thank you. Thank you again. Bye.